Gametogenesis refers to the process of formation and development of gametes, which are specialized reproductive cells. In humans, gametogenesis occurs in the gonads, testes in males, ovaries in females, and involves two distinct processes: spermatogenesis in males and oogenesis in females. Mitosis and meiosis are two different processes involved in cell division, each with distinct characteristics and functions. Here are the key differences between mitosis and meiosis. Purpose. Mitosis. The purpose of mitosis is the growth, development, and repair of somatic, body, cells. It produces two genetically identical daughter cells with the same chromosome number as the parent cell. Meiosis. Meiosis is specifically involved in the production of gametes, sex cells, such as sperm and eggs. It reduces the chromosome number by half, resulting in genetically diverse daughter cells. Number of divisions. Mitosis. Mitosis involves a single division of the cell nucleus. Meiosis. Meiosis involves two successive divisions of the cell nucleus, meiosis I and meiosis II. Chromosome number. Mitosis. The chromosome number of daughter cells in mitosis remains the same as the parent cell, i.e., they are diploid, 2N, cells. Meiosis. The chromosome number of daughter cells in meiosis is reduced by half compared to the parent cell, i.e., they are haploid, N, cells. Genetic variation. Mitosis. The daughter cells produced by mitosis are genetically identical to each other and to the parent cell. No genetic variation occurs. Meiosis. Meiosis generates genetic diversity through several mechanisms, including independent assortment of chromosomes, crossing over, and random fertilization. Stages. Mitosis. The stages of mitosis are prophase, metaphase, anaphase, and telophase, followed by cytokinesis, cell division. Meiosis. Meiosis I consists of prophase I, metaphase I, anaphase I, and telophase I, followed by cytokinesis. Meiosis II is similar to mitosis and consists of prophase II, metaphase II, anaphase II, telophase II, and cytokinesis. Resulting cells. Mitosis. Mitosis produces two daughter cells that are genetically identical to each other and to the parent cell. Meiosis. Meiosis produces four daughter cells, each with a different combination of chromosomes, resulting in genetic variation. First week of development, embryonic development. During the first week of development, the fertilized egg, known as a zygote, undergoes several important processes. Fertilization. The sperm penetrates the egg, resulting in the formation of a zygote. Cleavage. The zygote undergoes rapid cell divisions called cleavages, resulting in the formation of a solid ball of cells called a morolla. Blastocyst formation. The morolla develops into a blastocyst, a fluid-filled structure consisting of an outer layer of cells called the trophoblast and an inner cell mass. Implantation. The blastocyst implants into the uterine wall, allowing for further development and nutrient exchange with the mother. Second week of development. During the second week of development, the following key events occur. Formation of germ layers. The inner cell mass differentiates into two germ layers, the epiblast and the hypoblast. Bilaminar embryonic disc. The epiblast gives rise to the bilaminar embryonic disc, which consists of the amniotic cavity, formed above, and the yolk sac, formed below. Development of extraembryonic structures. The trophoblast differentiates into the chorionic villi, which establish connections with the maternal blood supply. The amnion and chorion also develop during this week. Third week of development. During the third week of development, significant changes take place. Gastrulation. Gastrulation begins, leading to the formation of the trilaminar embryonic disc composed of three germ layers: ectoderm, mesoderm, and endoderm. Neural tube formation. The neural plate, derived from the ectoderm, forms and undergoes folding to create the neural tube, which will develop into the central nervous system. Organogenesis. The process of organogenesis starts with the development of the heart, blood vessels, notochord, somites, and early formation of various organ systems. Spermiogenesis. Spermiogenesis is the process of sperm cell maturation in males. It follows spermatogenesis, which is the production of sperm cells through several stages of cell division. During spermiogenesis, the spermatids, immature sperm cells, undergo a series of structural changes to develop into fully functional spermatozoa, mature sperm cells. This process involves the following key steps. A. Golgi phase. The Golgi apparatus in the spermatid develops vesicles containing enzymes and proteins needed for the sperm structure and function. B. Cap phase. The Golgi vesicles fuse to form the acrosomal cap, a structure that covers the anterior portion of the sperm head. The acrosome contains enzymes necessary for penetrating the egg during fertilization. C. Acrosomal phase. The nucleus of the spermatid condenses, and the flagellum, tail, begins to form. D. Maturation phase. The excess cytoplasm is shed, and the mitochondria migrate to the base of the flagellum, providing energy for sperm motility. E. Final phase. The spermatozoon matures, and it is released into the lumen of the seminiferous tubules in the testes. Oogenesis. Oogenesis is the process of egg cell, ovum, development in females. It occurs in the ovaries and starts during embryonic development but halts at different stages until puberty. Each month after puberty, a group of immature egg cells is activated for further development. The process of oogenesis can be divided into three main stages. A. Oogonium stage. During fetal development, oogonia, primordial germ cells, divide by mitosis and differentiate into primary oocytes. These oocytes are surrounded by somatic cells called follicular cells. B. Primary oocyte stage. The primary oocytes remain arrested in prophase I of meiosis until puberty. Each month after puberty, some primary oocytes are selected to continue development. The surrounding follicular cells form a protective layer around the primary oocyte, creating a structure called a primordial follicle. Circa secondary oocyte stage. During each menstrual cycle, one primary oocyte resumes meiosis and undergoes the first meiotic division, resulting in the formation of a secondary oocyte and a polar body. The secondary oocyte then halts at metaphase II. If fertilization occurs, the secondary oocyte completes meiosis II, resulting in the formation of an ovum and another polar body.